Hello, everyone, and thank you for joining this webinar on the growing importance of translatomic research. I'm Michele Arnoldi, and I'm technical support and product specialist at Immagina. Here at Immagina, we are a group of people very passionate about translation, and we are based in Trento, in the north part of Italy, in the middle of the mountain. Here we have our headquarters, in which we do innovation, we have services part, and we do also production of the kits. But we also have a subsidiary in New York, and also a wide variety of distributor in all around the world. So the presentation of today is divided in 4 different parts. The first part is the introduction, then we have challenges, breakthrough technologies, and case studies. At the end of the talk, there will be a Q&A session, so you can just raise the hand in order to directly talk, or you can just type the question that you wish to ask, and we will read it and I will try to reply. Okay, so regarding the first part, the introduction. This is really a schematic on how translation works. I don't want to go into the detail. I just need to point out the main participant of the translation, such as the transcript, which encode information to produce the protein, the ribosome itself with the associated factors that enable him to perform its role of translation, the tRNA, and finally the protein produced. But let's go back a bit of history on what was in the past translation and how we reached this point now. So in the beginning, the tRNA and ribosomes were first discovered in the 1955. And since then, a lot of information were obtained. The first one was the first complete sequence of the tRNA alanine in 10 years later, in 65. And in 62, the RNA was also given a very important role because it was given that is responsible for decoding the information. And during those years, between the 60s and the 80s, a lot of different techniques were developed, such as sucrose gradient sedimentation, the SDS polyacrylamide gel, the IVT system, and the Western blot in general. All of those different technologies are still utilized today in order to study translation, and they were just the beginning of the journey. Since then, those technologies were utilized in order to study a bit more into that, the mechanism and regulatory pathway involved in translation. And in 1999, the Nobel Prize was given to the signal hypothesis, in which protein have a specific role, so they know what to do, where to be exported, or whichever information they are needed. And this was really an important path. Since then, the importance of translation and how to study the ribosome itself was proven way more into detail thanks to the cryo-EM and the study of the structure itself of the ribosome. Okay, so let's say to study translation in the beginning, the majority of the technologies were developed in order to study the final aspect, so the protein itself. Since then, a lot of different technology arise, both quantitative and qualitative, for the protein itself. But then a possibility to study translation was provided by actual understanding which transcripts are present in a cell, thanks to the sequencing of the RNA. So we can study how genes are expressed, if there are different isoform, and so on. But the actual bridge that connected two was provided in 2009 thanks to the ribosome profiling technology. So because it provides you a way in order to study which transcript are produced, which transcript are translated at that specific moment, providing also spatial information of where the ribosome was sit at the moment in which the technique was done. And for the future, the field is moving always toward a more approach towards single cell approaches, and there are a lot of effort in the latest year in order to go in this direction. It's the significance of studying translation is because it's very well linked to pathologies, such as in a neurodegenerative disease. You can study, for instance, the aberrant protein isoform of Huntington protein arise from expansion of the CAG repeat, but you can also study the accumulation of toxic protein in Parkinson's disease. Regarding cancer, you can move to study, for instance, tRNA, because tRNA leucine and tRNA tyrosine have been shown to be dysregulated in breast cancer. But you can also study the mRNA itself, because the MSXA modification have been resulted to be abnormal translation in colorectal cancer. Regarding infection disease, you can study translation because there are some pathogens that utilize alternative way of translation, like iris-dependent translation, but also they can change the way in which protein, especially host defense protein, have been synthesized. In cardiovascular diseases, it was shown that you can actually study some small protein called microprotein that have been linked to those pathologies. But overall, in general, the final goal is to find new therapeutic targets. So going back to technologies and approaches in order to study translation, as I was mentioning, the first one was given by the sucrose gradient, the technique called polysome profiling, in which thanks to a gradient of sucrose, you can separate the ribosome depending on their mass, and then you can fraction this different percentage of gradient in order to select specifically a specific subpopulation. Then you can then perform other technologies and other analysis, such as RNA-seq, microarray, or study the protein itself. It still requires specialized equipment and usually a large amount of starting materials, and is labor intensive. So these, since then, new technologies arise. Those are just some examples. It's not exhaustive, and there are more. But a possibility is to do trap-seq. In order to do so, you need to tag a specific ribosome with a tag that is recognized by an antibody. You can then pull it down and extract, for instance, the RNA to RNA-seq. But it's very difficult to construct those models, and you also need a very good antibody in order to pull down the specific tag that you inserted. Then, as I was mentioning, in 2009, ribosome profiling was discovered, in which you can utilize also spatial information, because you can have the information where the ribosome was sit, because you are going to produce individual ribosome thanks to a digestion, and the RNA that was embedded inside the ribosome that was at that moment translating can be retrieved, and a library can be done in order for sequencing. These still require specialized equipment and is labor intensive. So there was still room for improvement. In 2013, the RNA-seq was discovered. It's basically another way of using sucrose gradient, but in this case, you pellet everything together, so you don't have the possibility to isolate separately the different portion of the subunit and so on of ribosomes, but you pellet everything together. So you have a mixed analysis. As I was mentioning, the field, starting from 2021, started to work toward a single-cell approach in order to give a ribose-seq analysis to single-cell level. It's still relatively low in sensitivity and accuracy, so the goal is moving toward that direction. So in general, from the DNA information, the DNA genomic information, you transcribe into an RNA, you then translate in order to produce a protein. So why do we want and why do we care about the translatome part, not only the transcriptome part? Well, it's because it was proven to correlate better to the actual proteome compared to the transcriptome itself. So you can have a better proxy of the protein produced, still with an RNA-seq approach, studying the translation rather than the transcription itself. There are several reasons why you would choose to study the translation. So for instance, you can study how ribosome can associate with particular proteins, and here you have an example of SMN, in which you create a so-called specialized ribosome that can promote specific transcript translation. So in the disease, when you don't have this protein anymore, or you have a reduced amount, the translation of those specific transcripts is dysregulated, and this is linked to disorders. Here you have another example in which the incorporation of these pistol ribosomes are formed in order to respond to tumor immunity. So they are kind of produced to respond pretty quickly and to translate preferentially a subset of mRNA to do cytokine response. When this response is over, you don't need anymore those the incorporation of those pistol ribosomes is inhibited, and you can go back to a more so-called alert state. But as I was mentioning, ribosomes are not the only player in translation, because also tRNA can have a really deep impact, and you can play around with the anticodon stem and loop to regulate the actual gene expression, because you can alter decoding. So you can change stimuli from external stimuli in order to play around translation thanks to the tRNA. And this is sometimes linked to pathology state. So you have the ribosome that can have a role, you can have tRNA that can have a role, but we don't have to forget that you need the RNA itself in order to be translated. So you can actually play with the sequence of the RNA, translating RNA, in order to utilize, for instance, more optimal codon or higher secondary structure in order to increase the yield in protein production. You always have to keep in mind that if you play too much, you can still produce an unstable RNA that can be degraded thanks to the mRNA decay pathway. So you can have different ways in order to study translation, and depending on your need, you can find your your best way in order to study it. Another possibility is to study the mechanism of action of several drugs that goes directly to work on ribosomes, or you can find new translating transcript, such as the identification of new ORF, upstream, downstream, or microprotein that could be detected in those kind of analysis. And maybe they are more difficult to understand them and to study them with other technologies. So you can identify new novel operating frame with those technologies. Those are just few examples of why you should study translatomics in several different aspects of your research. There are still some challenges in to study this, and challenges in riboseq, the traditional riboseq analysis, is that it's time-consuming because you still need a ultracentrifugation step, so it's labor-intensive, and there is not really a protocol standardization because the different labs prefer to have their own homebrew technique. You can capture both active and inactive ribosomes because it's based on this fractionation that I was mentioning earlier, and usually required a large input amount. Regarding the tRNA analysis, there are not really commercially available methods, so there is not clear standardization. A lot of techniques utilize a reverse transcription part in which it introduces errors based on the different structure and modification that tRNA has. So these complex structures sometimes make issue in the analysis, and a lot of those technologies still utilize a gel purification step. So in try to overcome those issues, we developed several technologies. One is the ribolase, which try to overcome the limitation of the traditional ribosome profiling by avoiding using a ultracentrifugation step, thanks to magnetic beads and a probe that selectively pull down only active ribosome. I will talk about that later on. And these help out to reduce the amount of input material. For the tRNA-seq part, we internalize a technology called nanotRNA-seq, which allow to do direct RNA sequencing to study full-length tRNA, thanks to the Oxford Nanopore instrument. So it enables you to simultaneously study both quantity and which modification those tRNA have. So I just show you two technologies that we do have, but we do have in our portfolio a lot of different technologies in order to study all the different aspects that I mentioned in the initial slide. So the ribosome for ribosome profiling with ribolase and lase-seq, the tRNA with the nanotRNA-seq, HARIBO to study protein production, then a software, an IVT system, and the detection of small RNA fragment. You can find all of those on our website. For sake of time, I will just talk about riboseq and tRNA-seq. So for tRNA-seq, we do have this nanotRNA-seq kit, which utilize the technology developed in Eva Maria Novo's laboratories in CRG in Barcelona that enable to sequence full-length tRNA. So you can actually quantify their abundance because it's a direct RNA-seq, so it does not include any RT step. These allow to also detect modification. Now, the tRNA are highly modified molecules, so detecting their modification can be very useful for a lot of different analysis. And it's a gel-free method with a very fast workflow because it allows multiplexing multiple samples in a single run. So for the detail, the kit is very flexible because there is no species limit. The only limit is that you need to have the tRNA sequences. So the only limitation is to have the sequence of the tRNA. Then it starts with total RNA extraction, from which you isolate small RNA. Next, you deacylate the tRNA to remove the amino acid charge on them. There is then the library preparation step in which you add an adapter that is able to link both the 3' and 5' of the tRNA. Then there is the barcode adapter ligation in order to provide to each sample a barcode. And then there is a retrotranscription. I'm aware that I told you that there is no retrotranscription step, but now I'm showing you one. This is not the retrotranscription that canonically is done to then perform PCR in order to amplify a library, but is only provided in order to ease the complex structure of the tRNA. So basically, the retrotranscription is needed in order to let the tRNA to be to become a linearized structure, and then you have a hybrid RNA and DNA molecule. Since we added the adapter, then we can pull together up to 6 samples in order to run in the Oxford Nanopore machine. Now, in the Oxford Nanopore machine, in the pore, only the RNA will enter, while the cDNA counterpart will be eliminated. So it will enter only the RNA, and this is analyzed by the Oxford Nanopore machine. Then the signal is demultiplexed in order to have each singular library for the 6 different samples, and the data analysis can be done. So you can sequence on your desk. And everything is gel-free. Those are just some examples of some output that you can obtain, so you can actually count every single tRNA and understand how much they are expressed in this example, control versus treated sample. So you can do then statistics to understand if those fold changes are significant or not. But as I was mentioning, you can also understand which kind of modification those tRNA have. Now, the Oxford Nanopore machine provides a current signal. The different bases have a peculiar signal, but when they are modified, this signal is different. So you can infer which kind of modification you have based on changing in this signal. For instance, here in red, it means that in the treated sample, you have an increased modified current in this specific 37 position in glycine CCC. Now, which modification is this? Some modification can be predicted because are highly redundant, and it's based on the Oxford Nanopore way of understanding which modification they are. Some other, they need additional proof in order to be proven, such as mass spectrometry or other. But this tells you if in the control you have changing in which specific position. You can utilize this kit to expand translatome studies because quantified tRNA abundance and their modification can have an impact. You can also explore the ribosome-embedded tRNA because you can just collect the ribosome and then utilize this fraction in order to sequence specifically the tRNA present inside the ribosome. You can then perform colon-anticolon analysis and investigate the distribution in tissue and fluids. This ribo-embedded tRNA-seq kit will be released soon, and so stay updated. Regarding and moving on the ribo-seq gel-free technology, this kit comprehends two different technologies. One is the ribolase that enable you to start from either cell or tissue lysate and pull down actively translating ribosome thanks to our probe, which is a puromycin analog. I will go more into detail soon. Then you just extract the RNA, and the second technology that is together is the LaSeq, which is a library preparation that enable you to do next generation sequencing. Everything is without any gel extraction, and these provide a very streamlined workflow with 2 to 3 days in the lab. As I was mentioning, the lysate, the starting material, can be coming from various sample type, can be immortalized host primary cell, can be tissue. So far, the kit have been validated for eukaryotic sample. Now we are trying to move on to other specimens. So how does it work? Basically, from your lysate, you have a populational ribosome, the one just loaded on the transcript but not actively translating, and the one actively translating. Both undergo digestion in order to isolate those singular translating or non-translating ribosomes with the protected fragment of RNA inside it. This is a mixed population of ribosomes. Next, the idea is that we have this molecule called 3P, which is a puromycin analog. This puromycin analog is able to bind specifically actively translating ribosomes thanks to their conformation. So the idea is to functionalize a magnetic beads with this probe. Thanks to that, you can isolate actively translating ribosomes with just a magnet in order to remove completely the ultracentrifugation step. What I didn't tell you is that the nuclease that we utilize, when it cut, it inserts a signature which is a 3' phosphate. This modified group is the main part that is recognized by the library, the LaSeq library, because it internalizes specifically short fragment with a 3' phosphate, and the adapter is ligate on this specific signature. Now we can have multiple adapter in order to multiplex, also in this case up to 6 samples. Then this linked adapter and RPF with the 3' phosphate is circularized, is retrotranscribed and amplified by PCR in order for them to do next generation sequencing on Illumina platform. And then the reads are mapped and aligned, and what you can obtain is basically positional information of where the ribosome was sit on specific transcript. So the majority of the signal will be on the coding sequence because, of course, ribosomes are mainly translating on the coding sequence. But as I was mentioning earlier, you can maybe find something on non-translated region like 5' or 3' UTR, which might produce non-conventional open reading frame, or maybe you have ribosome read-through, and so on. Basically, reads are very short because the one protected by one ribosome are about 25 to 35 nucleotides in length. You can also have information of 2 ribosomes in very close proximity, the so-called disones, which have a length a bit longer. You can then separate those 2 populations, you can utilize those 2 together, and what you can obtain is for instance, this meta profile that basically tells you that the ribosome is moving 3 nucleotides at the time on a transcript, because of course the information is encoded in triplets. So the ribosome is moving 3 nucleotides at a time, and this is why you have this, let's say, jumping way of visualization of ribosome moving from the start to the stop of a transcript. Here is to show you that is very reproducible depending on the experiment that you're performing. And the main application is, of course, RiboSeq, but it's and to study translation start site, alternative open reading frame, translational control and regulation, but is a lot of time utilized together with the mRNA-seq in order to study translational efficiency, because you can have a transcript which is highly transcribed but not translated, or a transcript which is lowly transcribed but highly translated with the same number of reads, for instance. You can then understand whether you have a translational regulation rather than a transcriptional regulation by inferring the two information together. And this is a very powerful tool that a lot of people utilize. But let's see some, let's say, real-world application on the study of translation. So as I was mentioning, you can study several different aspects of translation. And in the beginning, I show you that you can study canonical or alternative translation to study different open reading frames, such as upstream, downstream, ORF, etc. But you can also study how the ribosome is translating and moving along transcript based on the difficulties that those ribosomes can have. In this example, if you have a stretch of lysine, well, translation can be a bit slowed down because then the rearrangement of the ribosome itself is trying to understand whether it's through this lysine stretch or is just a poly-A, for instance, a poly-A tail. So it just slows down translation. So you can study translational dynamic in this sense, but you can also study mRNA decay or recycling failure and so on. Another example that we saw is to study ribosome heterogeneity and specialization with the example that I did of an SMN-specific transcript. But overall, you can just study the development at several different stages, or whichever ribosomal synthesis defect you have, and so on. You can even study tRNA abundance and tRNA modification, rare codons usage, because overall, all these different modification and changes can change the pool of tRNA that are utilized. These will lead to ribosome stalling and ultimately alteration in translation. So you can play around with both techniques in order to have a better view of the translation in general. And depending on your needs, you can really understand better what's going on in your specimen. In biotech and pharma, tRNA have been studied because you can, as I was mentioning, play and optimize or modify itself, the tRNA, in order to optimize the protein production. You can enhance stability, specificity, and so on. But you can also utilize tRNA itself as a lead molecule in the tRNA medicine field to really study these as a possible therapeutics. In general, RNA and translation play a role in the RNA therapeutics field, such as the ASO and siRNA and so on. But there are especially in the last years, a lot of mRNA-based therapeutics that have been proven to be efficient, but you can play to inhibit translation, to enhance translation, and so on. So also biotech and pharma can utilize those technologies. In the clinical research, tRNA, and especially tRNA fragments, have been shown to be linked to several different, in this example, cancers. So you can study tRNA fragment as possible biomarkers and follow their availability in extracellular fluids, for instance, to study or to diagnose cancer, to study prognosis, or in general to understand how a treatment goes. This is just the beginning because the field is still pretty new, but this is another example of why it's important to study translation in general. So everything surrounding translation, in this case, tRNA fragments. The final goal in general is to go toward a more personalized medicine approach. So you can take patient samples and you can study all the different aspects of it. So you can do RNA-seq, ribosome profiling, tRNA-seq, mass spectrometry, and so on, in order to really understand more into deep which are the peculiarities of this patient and how to work better and to help it out better in general. So to conclude, I will just briefly show you few technologies that we have. We do have more. I explained to you why we do care about translation and why you could take advantage of studying translation for your experiment in order to increase the knowledge about your specimen in general. We do have other technologies and products, so please go on the website and check them out. And with this, I finished. So if you have any questions, just please write on the chat or just raise your hand and we'll let you in to ask the question. And thank you for being here today with me. I saw one raised hand. Okay. Hi, Andrea. Do you want to ask a question?
Yes. Can you hear me?
Yes.
Okay. Michele, thank you so much for the insightful and exciting presentation. I really appreciate a lot the tools you developed at Immagina. I have a lot of curiosity, but in particular, I would like to ask you a little bit about the riboless technology you explained. And in particular, the difference you said about the active and inactive ribosomes. So I'm not an expert about the creepy molecule and puromycin, but to the best of my knowledge, I know that puromycin mimics the tRNA entering in the A site of the ribosome. Is that correct? And so I suppose that for you, the active ribosomes are the one with the vacant A site tRNA. But for ribosomes in which the A site tRNA is occupied, for example, by another tRNA, or by factors like EFG or EF1A, or in a different stress like integrated stress response. Do you think that in that case that the ribosomes are completely hide by your technology and under view? And what is, in your opinion, a way to overcome this difficulty?
Mm-hmm. Thank you for the question. So in general, the probe mimics the puromycin, so the mechanism of action is slightly different. It does work similarly though, so you're right. If you have tRNA or in general something inhibiting the binding, this can maybe make it more difficult for the probe to pull down those specific translating ribosomes. Now, in order to overcome this, one of the possibility is to maybe pre-treat the specimen in order to let the tRNA exit completely, or any other factor that are present in the site to exit completely, in order to try to help out the probe to bind. In general, if the binding is precluded, yes, those population of very specific ribosome might not be correctly pulled down.
Okay, so in this case, even in presence of rotated or translocated ribosomes, in which the A site tRNA is in the AP site, in that case, if that part of the population is completely missed by the technology.
So the binding is not exactly the same. So if it's between AP, it might get pulled down. Now, how and where exactly it binds the probe, I cannot tell you, but it might be that some kind of conformation of the ribosome can still be pulled down, even in this situation.
Thank you so much, and congratulations again.
Thank you. I see that we have also a question in the chat. So Zuji-san is writing, thank you for the talk. I would like to ask if it's possible to detect RNA modification in your pipeline. So if you are referring to the tRNA part, hello, Zuji. So if you're referring to the tRNA part, yes, we can detect modification because it's based on Oxford nanopore. So it will provide a current change and the modification will be detected. There are some modifications which we know which kind of current they have because Oxford nanopore detected them, and they were trained for that specific modification. So those are easily detected. The other will just provide you a change in current. So you need to understand the modification with other techniques. So if you were referring to that specific, yes, we do have in pipeline the tRNA modification part. I don't know if I answer your question. Okay, thank you. Okay, so if there are no other questions from the audience, I would like to thank all of you for participating to this webinar, and I will wish you a good rest of the day.